We were talking about politics. No, we weren't. We were talking about memory on Tuesday, and I spent a little bit longer on some of the more cognitive aspects of memory than I intended to, and so we didn't get through all of Amnesia. So we ended by watching that clip from about Clive Wearing, right? Which I got to tell you was copyright flagged on YouTube, which is so annoying because how old is that? That clip is like 30 years old. And anyways, it's fine. You can still watch it. It's not a big deal. It was just funny that it got flagged. So we were talking about amnesia, right? And we were talking specifically about retrograde and anterograde amnesia, and how Clive Wearing had that viral encephalitis, which damaged his medial temporal lobes. So that's the first time medial temporal lobe has made an appearance. The second time the medial temporal lobe is going to make an appearance is when we talk about this guy, HM. Now, I've heard his last name pronounced a number of ways. Henry Molaison is how I've heard it from Stan Klein, who I learned memory from. But then there's a video that I'm going to show you about the mirror tracing task in a few minutes, where they say Henry Molaison. So, HM, right? HM. He was 27 when his entire life changed. He had intractable epileptic seizures. Like, they were not responding to any kind of treatment at the time. So this is the 1950s. They were not responding to any treatment whatsoever. And so last-ditch psychosurgery was to remove the origins, the starting point of the seizures. So they were able to locate through EEG where his seizures started. And they located them in the medial temporal lobes. And so he had the vast majority of his medial temporal lobes removed, which included parts of the cortex, mostly white matter, but some gray matter, and part of the basal ganglia that we're going to talk about specifically, which is the hippocampus. And the vast majority of those structures were removed on both sides. He had about he had full, I can't remember if it was right or left, but on one of the sides, it was full removal. And then on the other side, it was 75% removal. Okay. And after surgery, he was fine. He was operating normally, and he seemed to be lucid and everything like that. But very quickly, his doctors very quickly realized that he was unable to form new memories. Okay, so he had severe, profound anterograde amnesia. Okay, so we're like, something about what we just removed must be implicated in... Oh, poor Ellie. She's just having big feelings this morning. Must be implicated in memory somewhere. And memory formation, right? He also had some moderate retrograde amnesia. Various reports changed the numbers. 3 years, few months, that sort of thing. So anywhere between a few months and 3 years is kind of what he was missing. Maybe in that 3-year span, it was pockets. Not the entire 3 years, but leading up to the surgery, but pockets within that 3 years. And then, so that was considered moderate retrograde amnesia. And so that also harkens back to Tuesday as well, when we were talking about the time limitation for retrograde amnesia. It's very time limited. It's time locked, right? Because a lot of our memories get distributed elsewhere in the cortex. And so lots of Henry was still intact. Okay. But he couldn't form new memories. And so here's some, here are some images for HM. So this computer generated image was where, was removed. Okay. And so what they're showing you is the removal on one side compared to the non-removal on the other side, right? So we have removal, non-removal. Now, of course, he had both sides removed, as you can see in this image here. He had both sides removed, okay? So here's a posterior look at the remaining bits of the hippocampus, and then here are the anteriors. So this is back, front of the hippocampus removed. So the vast majority of the medial temporal lobe and this part of the basal ganglia was removed. Okay, so you can see, like, as we go from a more anterior, what kind of cross section are these? VIP status on the line. Whoever gets it first. Which cross section? Come on, you can do it. You can do it. I believe in you. I believe in you. I believe in you. VIP status. VIP status. Transverse. Transverse. We can do it. We can do it. You can do it. You can do it. Everyone can do it. Max wins. Max wins. Max wins. Max wins. Good job, everyone who was saying coronal, but Max got there first. VIP status coming your way, Max. You're gonna have a diamond next to your name, just like Jesse there. Yeah, see? That's what we do on this channel. That's how we do on this channel. Why? For no friggin' reason. But it's fun. And if you're rolling your eyes right now, think to yourself, what better could we be doing right now than just playing? So, all right, Max, you got that. I hope that makes you feel good. All right, you'll see that next time you're in chat, because I'm not going to do it right now. So we've got coronal. You would not have won. If you had said coronal, Jesse, then you would have been the first to say it if you had said it first. But you already have VIP status, so yeah, would have been. Then I would have just waited for the person who got it second. But very good, very good. Thanks, Max. I know you feel ecstatic. I know you love it. Don't lie. Don't lie. I know words don't carry intonation. All right, so we have these coronal cross sections at various slices of the brain. So here we're moving from an anterior. So this is so A is anterior, and D is the most posterior. So A, B, we're moving posterior to C and then to D, right? Okay, and so these coronal cross sections are very important because we're talking about very deep within the brain, right? So we have this anterior cross section where in the medial part of the temporal lobe you have the amygdala. So that's the first part that's the very front part. There's two little bulb-like structures on the basal ganglia at the very end of the big loop of the basal ganglia. Okay. So you can see that just above it is the entorhinal complex, or cortex, sorry. So that was removed as part of this psychosurgery, okay? The collateral sulcus was also removed as part of this surgery, okay? And so where we're talking about is where the hippocampus is, right? So we have the hippocampus here. You get rid of the hippocampus on this side, just to show you how this two structures are different, right? And then when we get to D, well, there's nothing that we need in D, and that's why here on this posterior view it was still left intact. Okay. Ollie and Murphy decided to come downstairs. Here comes Ollie. Hey, Ollie. Do you have a brain? Oh, Ellie's here too. No.
Yay! Hi,
Daddy! Hi! Hi! Yeah, do you guys want VIP status in chat?
Yeah.
Yeah, see, they're excited for it.
Hi!
Okay, you guys gotta head back upstairs. Okay, come on. Go. It's a race. It's a race. It's a race. All right, so that is the medial temporal lobe. And by the way, this is his head. By the way, this is his head. This is just an image of what was removed. This is actually his skull. He lived a long life. I think he died in the 90s. You can go look that up if you want on Wikipedia. But I think he died in the 90s. So he lived for like 40 years after this with no ability to form new memories, which is crazy. But here's the real kicker. Here's the real kicker with this. He was able to do a ton of tasks just as well as neurotypical folks with intact brains. Okay? And this is evidence that suggests, one, the medial temporal lobe, which is why we're talking about this now, is included in memory. But two, that it is separate memories from learning and specifically implicit or procedural learning. So one task that I want to share with you that he was tested on quite frequently is the mirror tracing task. Now, if you have time in all of your quarantined isolation boredom, I recommend this. What you do is you don't do it like this, because this is totally wrong. This is not correct at all. I'm going to show you a video on how to do it correct. You actually set up the mirror, and then you put something over the piece of paper, so you can't actually directly see your hand. Here you could see your hand directly, but it gets the point across, right? It gets the point across. So you are going to trace the inside of a star. So so you have an outer star line and an inner star line, and you need to trace with as few as errors as possible. What is an error? Well, going either too far inside the line or too far outside the lines. Okay. And so when you give this to a neurotypical person with an intact brain, they do it. They make errors in the beginning, but we get better at it. We can get better at it. So again, isolation. I want to see this on social media or TikTok. I gave didn't see any echolocation attempts, so I'm going to see mirror tracing attempts. I know you can do it. I know you have extreme boredom right now. You're bored. Stop with the sourdough. Stop with the baking. Mirror tracing. And then record yourself, just like this, every day. And then put it on TikTok for everyone to watch. Okay? It's my challenge to you all. VIP status for one that is shared with me. Okay? So if you have the ability to make a TikTok video of you mirror tracing, send it to me. I want to see the link. VIP status. All right. Mirror tracing. Now you can see on this graph, first day, these are how many errors. Okay, these are how many errors. So number of errors in each attempt. Okay, and so about 10 attempts per day. This is a neurotypical. This is a neurotypical response. Okay, so day 1, make a lot of errors, and then you get better by the 10th attempt. Day 2. You make some errors at the first attempt, and then you kind of just cycle around about 10%, maybe even less errors, right? So in or out of the line. By the 3rd day, pro status. 5% errors, a little line here or there. That little line, I wouldn't worry about that little line, that kind of thing. So neurotypical responses, right? This is what you would do in 3 days. I promise, I guarantee. Unless you are uncoordinated, for which I cannot help you, because that's a different problem. But what is also striking about this is also not a neurotypical. This is not neurotypical. This is HM. That's HM. This is HM right here. Day 3. That's HM What? I lied to you. I said it was a neurotypical response, and that's because it is, but it's not. This data right here reflects. I'm gonna grab it in my hands. That's HM. He can do it just as well as you can. Well, he could do it. He's dead now, but just as well as you. Just as well as you. Okay? Okay. Neurotypical, yeah. It's very interesting that you mentioned that, Julia, because neurotypical does include autistic people, actually. Because you have intact brains. Autistic folks have intact brains. Okay. It's much better than normal, because there's no such thing as normal. So, definitely not going to use that word. So here's the mirror tracing task. I was like, do I really want to? I want to break this up a little bit. So I wanted to show you all the mirror tracing task. So this is how you set it up for your new isolation boredom. Okay, this is how you set it up. Very simple to set it up. All you need is a mirror and a piece of paper and a marker and a thing to cover your hand. Okay.
We began this lesson by talking about Henry Molaison.
See, she said Molaison.
One thing we didn't discuss about HM was a task they had him engage in. In this task, neuropsychologist Brenda Milner, who was working with HM, had him perform a mirror tracing task like this one. HM was to trace within 2 lines of the star while viewing it only through a mirror, which was the reverse image. So if his hand moved one way, the hand in the mirror moved the other.
As mirrors do.
They asked HM to perform this task up to 10 times each day for 3 days. They counted the errors each time he went outside of the lines. What's interesting is that HM's drawings improved, even though he couldn't recall having done this task before.
So this is
looking at the times
her.
that I tried it.
Yeah.
And I'll be honest, this task was really hard to do. So what does this tell us about the brain? HM didn't remember engaging in this task, but he visibly improved. Well, it turns out that he couldn't form new explicit long-term memories due to damage to his hippocampus and surrounding areas.
I think
But
damage
he
is the
could
wrong
learn
word.
new skills, as was demonstrated by his performance on this task.
There you go. So that was the other thing that I was going to add, is he was getting better on that task, right? He was getting better on the task, but he would not remember a damn thing about doing the task. Doctors would ask him, so let's say day 3, he comes back to the lab. We're going to have you do a mirror tracing task. What's that? Never heard of it. Never done it before in my life. You sure? You were here yesterday doing it. Nope. I've never been here. You look familiar, but I've never been here. I've never done this task. This seems hard. You know things are reversed when you look at them in the mirror, right? And then they'd be like, give it a try. Give it a try. Okay. Hey, look, you did great. I guess I'm really good at it. Yes, you are, Henry. You are really good at it. It's day 3. That's neurotypical. That's what intact brain people are doing. Procedural or implicit memories don't need the medial temporal lobe. Okay. So we're going to talk about that a little bit more in just a few minutes, right? So I do want to bring up Alzheimer's disease specifically as a specific form of dementia, okay? And that is because it is... Oh, excuse me. And that is because it is important. For those of you who want to study elderly folks, it's important for knowing elderly folks because Alzheimer's diagnoses are on the rise. We still don't know a ton about it, but it is a memory deficit that is caused in the brain for something that's not just brain cells dying. There are it's quite a bit more involved than that. And you can see, so it's characterized by these amyloid plaques, beta amyloid plaques. So there's these dark things here, okay, these beta amyloid plaques. So they fill the cerebrospinal or interneuronal space, which is not good. because they're preventing your glial cells from doing their jobs. Glial cells are crucial to the survival of your brain because microglia, for example, are your waste glial cells, and they take care of all of the cellular waste. And so if you cannot get cellular material in and around the cells, then those cells will start dying. They will collect their waste, that will hang on to their waste. So microglia are really important. And then there are other glia that also have important jobs. But a society is only as good as its trash, its waste management, right? Rome didn't fall necessarily because it was attacked by Vandals, Visigoths, and all of the roaming tribes, right? It didn't fall just because of that. The internal structure of Rome was such that people were taking dumps in bathhouses where the sewage just sat underneath. Disease, right? British scientists believed that cholera was carried through the air because it was so polluted. No, it's in the water that you're drinking. So society is only as good as its waste management. And if you can't get glial cells, and yeah, lead plumbing, if you can't get glial cells to manage the waste inside the intracellular space, then you're going to have problems with those neurons. The other issue with Alzheimer's are neurofibrillary, it's a tough word, neurofibrillary tangles. So these are fibers that become twisted around the soma and axons, sometimes dendrites, but the larger structures of the neurons with the tau protein that is called tau, T-A-U. Okay, and so they build up and they prevent cellular transmission. And so we have amyloid plaques that prevent intercellular business as usual. And then we have neurofibrillary tangles that prevent intraneuron things. And so what ends up happening is those neurons begin to die. You end up having pocketings. So cavitation is so if you were to exhume and do an autopsy on a severely advanced Alzheimer's patient, and this has been done extensively, then you will see pockets in that person's brain. Okay, the holes, and those holes have been caused by these suckers. Okay. So Alzheimer's, I think, and so the cause of Alzheimer's is really unknown at this point. There's potentially a genetic component. There's likely an environmental component. There's likely a psychological component. So it's as far and most of the effects you see are memory related. Okay. And there are plenty of great films that explore this. So as much as The Notebook is annoying as far as a love story goes, it's a really great depiction about using various things to help somebody with their memory. So what else? Oh yeah, Still Alice. It was between The Matrix and Still Alice. To do Matrix. I had The Matrix, so that's why I ultimately chose it for the class film. But Still Alice is a really good depiction of somebody who is succumbing to early onset Alzheimer's. So it's somebody's 40s or 50s, early onset. Most Alzheimer's patients generally start seeing the effects in their 70s and 80s. Early onset is like around 50. So that's not good. Okay. All right. Any questions about memory deficits before we move into the neuropsych of it all? and specifically the neuropsych of long-term memory and the divisions of long-term memory. We're not going to do short-term or sensory memory from the model. If there are any questions, I'm going to take a drink. Okay. All right, long-term memory. This is the stuff you know. As I mentioned on Tuesday, it's seemingly infinite, seemingly lasts forever, and its capacity is seemingly never-ending. Sure, you can forget things. That's probably just because you can't find it anymore, right? So we're going to talk about the neuropsych of these divisions here. So the first one that I want to talk about is your verbal, your explicit, your declarative memory. So as the video said with the mirror tracing task, these are your semantic memories, these are your episodic memories, these are the relationships you have amongst that information. And the reason why it's called declarative is because I can think about something that's happened in my past and verbalize it. Okay, I can verbalize it, I can talk about it, I can explain it, I can describe it, I can put a time stamp to it, I can put a space to it. Time and space are really crucial for declarative memories. They are memories that are a part of you that you form every day. Okay, that is characterized then, that is then contrasted. That's what I want to say. Contrasted with nonverbal, implicit, or non-declarative memories. These are memories that you learn that information. You can then use that information because it's a past experience. It's information from the past, but you cannot verbalize it. You cannot declare it. You cannot describe it. It's very difficult to tell somebody how to do something, right? And so we've got priming, we've got conditioning, we've got habituation and sensitization, we've got procedural memories, skills. It's very difficult to tell somebody how to gain a skill rather than to show them how to gain a skill. You actually have to engage in doing a skill in order to get the memories for that skill, okay? And so if we go back to Clive Wearing, he had those convulsions going back and forth between these two that you saw at the end when he was playing the music and he was singing, because that was his brain using this information, but it was traveling through the areas that were deficited, that were lesioned because of his viral encephalitis. And so that would cause convulsions because this intact information was being routed through areas that were not intact, okay? And those were his, very appropriately, his semantic and episodic memories. But again, you saw that in Clive Wearing's case, he does have time-limited retrograde amnesia. He remembers things about his life. He remembers Deborah, obviously. He remembers his kids. He doesn't remember that his kids are older, or were older now, or are older. Of course, they're growing. He remembers them as little because he can't take the new information of them growing. Okay. And so all of this stuff is the stuff that's not intact, but over here, that stuff is intact. Okay, so before we talk about this stuff, I want to talk about the neuropsych of verbal, declarative, explicit memories, right? And that is, excuse you, there we go. That is the medial temporal lobes in all of their glory, okay? So from HM through monkey lesion experiments, so this is a monkey brain that you're looking at, all the way through Clive Wearing. You heard the doctor in the clip say the viral encephalitis attacked his temporal lobes, and specifically his medial temporal lobes, and basically ate through. The virus destroyed all of those areas, right? And so medial temporal lobe, all the evidence and information points to the medial temporal lobe as the central location for new memory formation, and specifically new explicit memories. Okay, and the structure that's involved in that specifically is the hippocampus. So here we have this monkey brain. And so the hippocampus is outlined in blue. This is supposed to be medial to the so it's not very well defined here, but these two structures, the amygdala and the medial temporal lobe, are supposed to be, or excuse me, the hippocampus are supposed to be embedded within. That's why that's called medial, right? And so they're shining, they're three-dimensionally shining through this lateral view of the left hemisphere, right? And so this is duplicated in the right hemisphere. So if we take a Katerina, just to clarify, the hippocampus is the medial temporal lobe. Yes, it is. The basal ganglia is three-dimensional in the center of the brain and goes then into the structures of the temporal lobe. Yes. So these structures are defined within the white matter of the temporal lobes. Okay. And so if we take this posterior view, or ventral view, actually, since we're talking about the cortex, this ventral view of the brain, so we're looking at it from the bottom. So this is the front, and this is the back. Okay. You can see how they then three-dimensionally come through the inside of the temporal lobe. Okay. So they essentially are hugging the ventricle and thalamus of the so you have the diencephalon. The thalamus comes up as two fists, kind of like this, the right and the left thalamus. You have ventricles surrounding that, and the hippocampus inside the medial temporal lobe hugs just the outside of that. Okay, as the basal ganglia comes around it. Okay, I really wish I had Brian right now. Okay. And so with monkeys, then we can do different tests, because you can lesion. Unfortunately, you can lesion monkeys and see whether or not the evidence of HM can be replicated elsewhere, right? You need to be able to replicate it. Because what if HM was just a unicorn of psychology? If he was just a unicorn, took out his medial temporal lobes, he couldn't form new memories, boom, unicorn. No other medial temporal lobe lesions led to the same effects. That would be real bad. Because we wouldn't have replicated evidence of the function of the MTL. And so monkey experiments are crucial to replicate the effects seen in HM, because we can lesion these monkeys. Okay? Because of reasons, right? Animal testing. So, one of the ways that researchers confirmed the MTL in memory, explicit memory, was doing these monkey experiments with what's called delayed non-match to sample. So that's the methodology, the name of the task, delayed non-match to sample. So let's take this one at a time as far as the methodology goes. So so there are two bins here, and you can see that one side the experimenter's on, the other side the monkey is on, right? So we have two bins where food is, okay? And so food is used as a reinforcer because it's rewarding, it's probably yummy and delicious. A cue is placed over the food bin, okay? Then the monkey, the shade is lifted, and the monkey has to choose the correct response. It's basically shown the correct response. it yields that reward for that human being, right? That human being, I don't know why I said human being. It yields the reward for the monkey. Okay, so that's in B. Okay, Q is the cross. Monkey picks it. Reward. Yay! Shade is closed. Let's go to C. Shade is closed. There's a delay. Okay? There's a delay. The reward is placed in the other module, and a new Q is placed over it. It's just a black square. Instead of a white cross, it's now a black square. Okay? After a period of delay, the shade is reopened, and the monkey then needs to choose which Q has the food underneath it. A monkey who does this a lot will be good at it. Okay? An intact brain monkey will be good at it. Okay? If you lesion the monkey's medial temporal lobe, the monkey will not be good at it. If you lesion the prefrontal cortex, the monkey will be okay. Because you're only damaging the working memory, and because you've added a delay, they're good at it. If you lesion the medial temporal, so again, delayed matching to sample. Let me put that, there we go. So delayed matching to sample. And you can delay 8, 15, 60 seconds, right? So 8 seconds, monkey's fine, regardless of the lesions. But if you destroy the hippocampal and parahippocampal cortex, HP, amount of delay drops it off typically. N stands for neurotypical monkeys, or normal monkeys, intact monkeys. They only get as bad as about 85%, even with a delay of 600 seconds. Delay of 600 seconds is how long? That's 10 minutes, right? It's a long delay for this kind of task. But look how much that drops off for destroying the hippocampus and parahippocampal region, okay? And even further drops off this blue line here if you destroy not only the hippocampus, the parahippocampus, but also the pararhinal cortex. So all three, look how much more it drops off, even after 15 seconds of delay. So comparing these three at 15 seconds of delay. If you lesion the prefrontal cortex, it doesn't really matter. So therefore, the lesion studies give us a double dissociation. Medial temporal lobe is for long-term learning. Prefrontal cortex, not for long-term learning. Maybe for working memory? Maybe. We haven't gotten there yet. Okay, well, we're actually not going to talk about that. I think your book talks about it, but we're not, right? So we have a double dissociation on lesions to these monkeys, and the medial temporal lobe is for long-term learning, especially when we damage all of those regions here, HPP. Okay. Let me know if you have any questions about that. What did I... Oh, yeah. I like this one because it's showing you where all of these places are in mammalian brains. And so, of course, our brain is most connected, are most similar to a primate brain, because we are primates. And then you can see how then this is connected to a rodent brain, right? So which is a lot smoother, a lot smaller, right? There's not a lot of convolutions in a rodent brain, but you can see where all of these regions are connected in those rodent brains. By the way, so this is the anterior portion of a rodent brain, and this is the posterior portion, or rostral caudal. Okay, so just so you can make it out in those regions. Okay. Oh, what else did I want to say about this? Not much. Not much. It tells you how it distributes the information, or the information gets distributed, but you can basically say, oh, that's just cortex. Okay? That it gets distributed to the cortex. Hippocampus, cortex. Okay. So I want to then just now jump into, so now that we have evidence for the hippocampus being really important in forming new memories, I want to talk about encoding in it, and then I want to show you a really cool aspect of how this works in real life. It has to do with London taxi drivers. So I have this clip to show you in just a minute. So let me tell you first about this task that you see here. This task. Oh, yeah, that one with the graph on it. Okay. So this one was done with humans. This was a task that was done with humans. So in A, up here, the design of the experiment is quite important. Okay. So that in a scanner, although the for some reason your book does not have the scanner results, so we're just going to have to look at the behavior results. So at encoding, so this is the learning phase, participants viewed a series of words and made either an animacy judgment, so whether or not the word represented something animate or inanimate. So here we have nickel, right? A nickel is inanimate. Or they made a size judgment. So they were given large or small buttons. So is something large or is something small? Okay, depending on that color. So the color of the word indicated which judgment they were supposed to make. So green represented animacy, and red represented large or small. Okay, so nickel was in green, and so they made inanimate animate. And then deer is in red, and so they made large, small. You could say that was large, right? And so then they did a test phase. Okay, so they learned these words, and then they did a test phase. And so in the test phase, they showed the same words they saw during the encoding phase, but then also new words. They made the same two judgments, large or small. Okay. Large or small. Then they had to indicate how confident they were in their recognition of the word. So not just this was a new word or this was an old word, but whether they were confident. Okay. So that was on a scale from 1 to 6. Okay. And then they had to say if they saw the word as previously red or previously green. So they're in the test phase they were doing 4 judgments. If it was green, or actually one of 3, they were making 3 of 4 judgments, I should say, because each word then was either only one color. So the first 2 is part of the 3. So was it green? Is it animate or inanimate? Or is it red? Is it large or is it small? So that was the first one during. So they same thing as the test, as the encoding phase or the learning phase. So during the test phase, they did one of those 2. Then they did 2 additional. How confident are you in recognizing the word? Did you see it before? On a scale of 1 to 6. And then what color was it in during the encoding phase? So animate and inanimate, green. Large, small, red. How confident you? 1 to 6. 6 being the most confident, 1 being the least confident. Was it red or green during the encoding phase? And so you see here are the behavioral judgments on new versus old words. Okay. So this is a classification, not from the judgment that the participant made, but the classification from the experimenter's perspective, okay? So we had definitely new and definitely old, and this is confidence. So how confident are you? 6 This is definitely an old word. I'm super confident that it was old, that I saw it before. 1, this is definitely new. I had not seen it before. It's definitely new, right? And then so the y-axis is the percentage of total words. And you can see how much they change, right? So lots of blue over here for new words, and lots of green over here for old words, okay? What you don't see is where this activation is occurring, and that's in the hippocampus. That's in the hippocampus. And it's specifically very important that it correlates with this part of the scale, the green behavior. Activation in the hippocampus is part of the green behavior over here. More activation over here, less activation over here. More evidence. So let's take a bit of an interesting break here of me talking, and let's talk about how London cab drivers are amazing human beings that remember just everything. And what is striking about this, and I don't remember if it's in the clip or not. It's been a couple of days since I've watched it. I don't remember if they talk about the brain size changes here. But McGuire and colleagues in 2006 actually did structural scans of London cab drivers versus other people, non-London cab drivers. And they found a significant size increase in London cab drivers. And here's a taste of what they actually have to do. No! I thought that was going to work. That's annoying. That is really annoying. All right, let me find another one. I'll be right be right back. I'll be right back. Well, I mean, I'm going to be here. Ask any questions. I'm going to go find a new one so we can actually watch something, because it's really, really interesting. Jerks who took it from me, who stole it from me. London cab driver hippocampus. All right. Well, obviously can't watch that one. Do I want to do National Geographic? AFP News Agency. Get out of here, you jerks. All right, we'll watch this one. So funny. All right, we'll get rid of you, and we will insert here a new one, a new video, online movie, Insightly. Put it back over here. Sorry, everyone, for the brief interlude here. I'm going to go ahead and save it. Okay, coming back online here in just a second. Let's go back into this mode. Let's go back over here. Back over here. Here we go. Let's watch this one. Somebody did a screencast.
London's legendary taxi drivers are the Olympic athletes of memory. Navigating
so you can see me moving
central London,
smoothly.
They must create an
since this is a screen recording of a video,
streets, landmarks, and locations.
It's going to feel choppy. It's going to look choppy.
University of London scientists studying the shape of memory found ideal subjects right in their own backyard. For 150 years, every new cab driver has had to pass a grueling exam, known to Londoners simply as the knowledge. It takes most hopefuls at least 2 years to master, giving researchers a superb opportunity to look at how the brain adapts when required to retain vast amounts of information.
Thanks, mate. Can you take me to Albert Hall? Albert Hall, yeah. Cheers.
When somebody gets in your cab, they'll say, take me to so-and-so, and it's got to be like that. You've got to know instantly where you're going, which way to be pointing. So there's a lot of retention. You've got to retain a lot of what you've learned as well.
Scientists suspected a particular region of the brain called the hippocampus might be the key to the taxi driver's success.
Now, run me to the
that was an
nearest
awful
police
representation.
Waterloo Bridge, Ford Lancaster Place.
If memory depends upon the hippocampus, at the Knowledge Point School, drivers in training surely give this part of their brain a good workout.
Dave, give me the name of a restaurant on Portland Road with a lady's name.
Chutney Mary's.
Hereford Road, where would you give me?
Veronica's.
After class, students like Andy Miller take to the streets. Since visual processing occupies more brain activity than all the other senses combined, it is not surprising that direct experience is an essential part of the knowledge training.
You start off learning all the roads, then you have to learn all the places on every road. With all the routes that you have to do for the knowledge, you couldn't possibly do it on a map. You have to get out on your bike in the rain, the cold, the snow. You learn it bit by bit.
But how does the brain retain and order all that information? As the scientists suspected, mastering the knowledge may have a physical impact on the brain itself. Their study concluded that part of the hippocampus was indeed larger than average in these drivers. In fact, the most dramatic differences were seen in the drivers who were on the job the longest.
The hippocampus has a spatial map in it, and what seems to be happening in the taxi drivers is that the spatial map is laid down of central London, and laying this down causes the connections to develop and grow and more of them to form, and that makes part of the hippocampus get bigger.
It's almost like you've somehow, somewhere up in your brain, you've created enough space to sort of slip this map in, a little bit of software.
This study suggests that the adult brain can refashion its basic anatomy according to the requirements of its owner. Doctors hope these insights can be used to help restore the minds of stroke victims, Alzheimer's patients, and those suffering brain traumas.
All right, questions about that. I know a question. I have a question. Now that we have, like, these things, is this useful anymore? Do we care? Do we care about London cabbies? That's a question for you all. Take a London cab? Take an Uber? What if you get horrible service? Here's the thing. Yeah, London cab drivers are still doing that. They're still engaging in that knowledge point school. They're still... to be a London cab driver, you still have to do that. You can't just go, okay, I'm going to plug it into my GPS and go, bleep, 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 blah, 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 blah. Okay, cool, great, awesome. You can't just do that. You have to, these London cab drivers still have to go through this training. I can amaze you the next time you're in London. Okay. So this is so Eldrigan and colleagues. This is further evidence that the hippocampus is important. Most modern studies will be brain imaging studies, so that sort of thing. Alabama man, no, I did. I mean, I streamed. Streamed review sessions with my students, but I did not teach classes before the COVID stay-at-homes. This was a directive that came from the school. Well, governor to the school, I suppose. Yeah. The medial temporal lobe is useful with familiarity as well. Okay. And recollection in general. Familiarity, medial temporal lobe activated, but then there is other areas on the cortex that are included. So a lot of the frontal area is required for recollection on both sides. So that's a lateral thing. So cortical activation in recollecting things that have been acquired, recollecting later the things that have been acquired. And so not only is that evidence for the medial temporal lobe being important and the hippocampus being important for memory encoding, but these explicit things like I can recall a memory or either an episode or a fact from the top of my head, from my head. Those explicit things, hippocampus is generally important for, but it's also evidence that suggests that our cortex is used primarily for the storage of all of these memories to be used later. So we just basically we encode it in the hippocampus, and then it gets consolidated and branched to other places on our brain. On our brain. That's what I wanted to say. Retrieval. You can see here that the anterior parahippocampal gyrus and the bilateral hippocampus are useful for retrieving. So not only do we need to encode those memories in the hippocampus, also the hippocampal regions are used for then bringing those connections back to consciousness. You can think about them as two-way streets. So new explicit memories get formed in the hippocampus. Those get distributed to the cortex. But if you need to retrieve something, those same connections then bring them back to the hippocampus for retrieval. Okay. Again, further evidence of this from Montaldi et al. So a lot of this stuff you can read more about in the textbook. I'm just going to move on from that. So we can talk about implicit memory for a little bit. So here's classical conditioning. So this is learning without the hippocampus, which is great. You can create a situation where a conditioned stimulus, like this bell, causes a conditioned response, salivating from the dog, right? So that's part of implicit memory. The dog has no recollection of what the bell means versus what the bell is doing. Okay, just same with you and all of your classical conditioned responses, like a loud siren in your ear is going to make you startle. So good startle response. You don't actually have to remember where you learned that, when you learned that, who you learned it from, when you first did it. You probably first did it when you were like a day old. And so you don't need the hippocampus to do these things, because we saw that dissociation with HM and the mirror tracing task. Okay, not necessarily classical conditioning, that was procedural learning, but be that as it may, it's stuff that doesn't need the hippocampus because he didn't have his hippocampus, right? Procedural memory also. So people were given this 4-finger response task, and you had to learn the sequence of lights. It's kind of like if you've ever played the Bop It, right? Or Simon, right? It's called Simon, the 5 lights, and you had to remember the sequence, or like Bop It, which was like doing twist it, bop it, throw it, shoot it. Yeah, bop it. Okay, so it was a situation like that, and you press the 4 buttons corresponding to the 4 lights, and you had to use all your fingers, right? Even your gimpy finger, your pinky, which is useful for nothing other than maintaining fine motor movements. Okay, and so you had to press buttons with your pinky, which is, I don't know, difficult for me to do. For those of you who may be better typers than me, may not be. Astrid holds her pinkies out. Well, actually, no. She holds one of her pinkies out when she's typing, and she holds the other one in. It's very strange. Shh, don't tell her I said that. And so you can see that as a person does this, they get better at it. That's what this curve is showing you, right? So when you first start doing it, oh, you're so bad at it. The sequences, like 1, 3, 4, 2, 1, 4, 2, 3. I can't press that fast. Please stop. But it's just like with the mirror tracing task. The more you do it, the more you engage in the task as a skill, then you get better at it. And that is that. If you still have a Bop It, again, coronavirus lockdown fun. Play Bop It. Play some Bop It. Yeah. I think there was a recent like SNL or something like that where they made a Bop It that was supposed to be really, really bad, like crass and gross. And I think Beck Bennett was like, "Wait, I have to do that." Or wait, what's this? And it was like a dad, and he was like addicted to it because it kept telling him to do stuff. Kill it, murder that. Yeah, it's good. I think that was recent. I think that was a recent SNL thing. All right, last thing, and then we are done with memory. Is the connection between all of these areas. So if we're talking about the anterior versus posterior, so the top versus the bottom, top versus the bottom. So not top versus bottom. It's front versus back. So in red is the front, back is in blue. Okay. So like we've been talking about with dorsal and ventral streams, memory does have the has like 2 locations for processing, right? So memory processing happens in the anterior portions, okay? And so that's earlier stuff, okay? Earlier stuff. And then later stages of processing happen over in these systems. So anterior system, posterior system, okay? So there are 2 memory systems, all right? Any questions on memory either today or from Tuesday before we end with this material? Bop it. Twist it. Bang it. Launch it. Twirl it. If you have seen, I'm slowly going through the second drafts. If you haven't gotten one in, please get one in so I can do it. And then, so that should be done at some point next week. And then the film analyses will be graded at some point next week as well. Excuse me. Any questions? What are you all doing this weekend? I'm going to go ahead and add the recording there. I'll stay on the stream for a sec just in case there are any questions.